Welcome to Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we're understanding the mechanism of action and the specificity of enzymes in terms of their three-dimensional structure. We're also understanding that enzymes are biological catalysts that reduce activation energy. And finally, we'll look at that there are intercellular enzymes catalyzing reactions inside cells and extracellular enzymes by cells catalyzing reactions outside of the cells. So what are proteins? Proteins are made through the process of transcription in the nucleus and translation in the cytoplasm. And proteins are constructed through different stages called the primary structure, secondary structure, and tertiary or quaternary structure, depending on the protein being made. The primary structure of a protein is determined by the gene sequence. There is a possibility of mutations happening in this stage, such as an incorrect gene or gene sequence being generated, and this can affect the tertiary structure that is produced. So now we're looking into enzymes. So enzymes are a type of proteins that are biological catalysts that catalyze metabolic reactions. Their tertiary structure determines the shape of the active site. They are very vital in the human body and are involved in important functions such as collagen production and respiration. Enzymes can be of two types, intracellular and extracellular. So intracellular enzymes catalyze reactions inside the cells, while extracellular enzymes catalyze outside the cells. Enzymes have an active site, which is where the enzyme binds to a specific substrate to catalyze reactions by lowering the activation energy. This binding of the enzyme to the substrate is called the enzyme-substrate complex, and enzymes are called specific as they only catalyze one reaction. Different enzymes have different tertiary structures, resulting in different active sites and making them specific. Need to reach the activation energy for it to take place. In simple terms, activation energy is the energy needed to start a reaction. Enzymes are beneficial as they lower the activation energy, and they make reactions happen at a lower temperature. Looking at the active site. So if substrate molecules need to be joined, being attached to the enzyme holds them close together, reducing any repulsion between the molecules so they can bond more easily. Alternatively, if an enzyme is catalyzing a breakdown, binding to the active site will put a strain on bonds in the substrate, hence breaking the molecules more easily. So sometimes the purpose of an enzyme is to join two molecules, and sometimes it is to split them apart. And the first bullet point refers to when you're joining two molecules using an enzyme, and the second bullet point is talking about splitting a molecule into two or more molecules using an enzyme. So, however, because of extreme conditions such as change in temperature or pH, the tertiary structure of the enzymes might change, resulting in the change in the shape of the active site, hence the reaction no longer taking place. This is because the substrate no longer fits the active site, resulting in no enzyme-substrate complex, hence no longer being able to perform their function. Looking at some enzyme models that we need to know. So the first one is the one you're familiar with, the lock and key model, which is the theory that enzymes are specific to the substrate that they bind to. There is new evidence that suggests the possibility of a new theory called the induced fit theory. This suggests that the enzyme substrate complex changes shape so that it would fit perfectly. And this locks the substrate more securely to the enzyme. More enzyme molecules are present in solution, the rate of reaction will increase as there are more active sites present, as the probability of enzyme substrate complex forming will also increase. However, after a certain period, the rate of reaction will level off as the substrate is the limiting factor, as there are no more substrate for the enzymes to bind to. The point where the rate of reaction levels off is called the saturation point. So if you plot this on a graph, the point where the actual graph begins to level off is going to be your saturation point. And it's quite important to know what the graph looks like for rate of reaction compared against enzyme concentration and rate of reaction compared against substrate concentration. So the graph is going to be very similar, where there's going to be a steep increase initially and it's going to start leveling off after the saturation point. Similarly, an increase in substrate concentration initially increases the rate of reaction, and this is because the possibility of enzyme-substrate complex increases and more active sites are likely to be used. However, after a certain point, just like the comparison of the rate of reaction with enzyme molecule concentration, the rate of reaction levels off as the enzymes are the limiting factor now. The reaction has reached the saturation point. So this is when the substrate is no longer the limiting factor, it is the enzymes. Hence, there are no more active sites to bind to. Over time, the rate of reaction will begin to decrease if none of the variables are available. That is all from Professor Snap today. Hope you found this topic useful. If you have any questions, let me know in the comments below and I will get back to them as soon as possible. And don't forget to like, share, and subscribe.